All living things on Earth are made of genes. Little pairs of A's, T's, C's, and G's that make us who we are. Even one letter changing in the entire genome of 6 billion base pairs can cause horrible, if not fatal, diseases. With many diseases, we know exactly where in the genome this mistake is. So what if we could figure out a way to get to that spot in the genome and change it back to what it's supposed to be? That's where gene editing comes in, and the most exciting form is CRISPR-Cas9. It's making headlines all over the world, but how exactly does it work? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is found in many different types of bacteria as a way to defend from viruses. The DNA sequences in CRISPR consist of short sections that are the same forward as they are backwards, for example, ACT, TCA. And between those sections are sequences that correspond to viruses that the cell has encountered before, as a way to remember how to beat those viruses. The Cas9 element is what then carries out the destruction of familiar viruses. Cas9 is short for CRISPR-associated protein, which acts as a nonspecific endonuclease, which is scientific jargon for a pair of molecular scissors that can go and cut genetic material. The Cas9 protein uses a short fragment of guide RNA, also known as gRNA, and is able to match it to the sequence that it's trying to degrade so that it knows exactly where to cut. Let's see what happens next with this cool pair of scissors. The scissors represent the Cas9 element, and let's say I want to edit this sequence of DNA. Here I've fed it some guide RNA that corresponds to the target sequence. So you can see that the sequence is exactly the same as the sequence that I want to cut. It matches perfectly to the leading strand. Once it's there, it will bind the DNA right at that sequence, which I can do with this cool little paper clip, and that lets the Cas9 element know exactly where to cut on either side, and it creates blunt cuts. Once that sequence is removed out, the cell naturally has a repair system to take care of any cuts that might occur in the DNA. And so that can either join the two ends together, which might add or remove a certain base, or it can add in a new sequence, which the scientist provides, and add that in, effectively adding new DNA to the code that either codes for the correct sequence, or it could even be something completely new. CRISPR can be an incredibly powerful tool for tackling genetic diseases and conditions that we otherwise wouldn't have any way to deal with. There are risks associated with it and a couple ethical concerns, but as research continues, we can hopefully develop it so that it can help save lives. Thanks for watching.